Hey everyone, hope you're doing well. Welcome back to our IGCSE Biology Revision Series. Today, we are diving into Chapter 5, one of the most important topics in biology, enzymes. By the end of this video, you will understand what enzymes are, how they work, and why they are absolutely essential for life itself. Let's get started. So, what exactly are enzymes? Let me give you three key facts to remember. First, enzymes are biological catalysts. The word biological means they are made in living cells, and catalyst means they speed up chemical reactions without being changed or used up themselves. Second, all enzymes are proteins. This is crucial because being proteins means their structure is absolutely vital to their function. Thirdly, enzymes are essential for life. Without them, the chemical reactions in your body would be far too slow to sustain life. Here's a mind blowing example. If we didn't produce digestive enzymes, it would take about 2 to 3 weeks to digest a single meal. With enzymes, just 4 hours. That's the power of enzymes. Enzymes maintain the reaction speeds of all metabolic reactions. That's every single chemical reaction keeping you alive right now, at a rate that can actually sustain life. From breaking down your food to building new proteins, from respiration to DNA replication, enzymes control it all. Let's explore the lock and key model first. Think of an enzyme like a lock and its substrate as a key. The substrate is the molecule that gets broken down or joined together in the reaction. Every enzyme has a special region called its active site, and this is where the magic happens. The active site has a very specific three-dimensional shape that is complementary to the substrate, meaning they fit together perfectly like a lock and a key. In the lock and key model, the active site is already the perfect shape for the substrate. It's rigid and unchanging, just like the lock waiting for the right key. But here's the thing, the lock and key model is just a simplification. Modern research has shown us a more accurate model called the Induced Fit Model. Let me walk you through this step by step. Step 1 is the initial approach. The substrate approaches the enzyme's active site. Here's the key difference. The active site is not a perfect fit yet. It's close, but not exactly complementary. Step 2 is where the substrate binds. As the substrate gets closer and begins to interact with the active site, something amazing happens. The presence of the substrate causes the enzyme to change shape slightly. The third step is the induced fit, where the active site changes shape. This is a crucial moment. The enzyme is flexible, not rigid. As the substrate binds, it induces the active site to change the shape and mold around the substrate. The enzyme literally wraps around the substrate to achieve a perfect fit. The active site adjusts its shape to become complementary to the substrate. Step 4 is the enzyme substrate complex formation. Now we have a perfect fit. The enzyme and substrate are bound together tightly, forming an enzyme substrate complex. This tight binding puts strain on the bonds within the substrate molecule, making them easier to break or helping new bonds to form. Lastly, the catalysis occurs. With the substrate held in exactly the right position and the right bonds under strain, the chemical reactions happens much faster. The substrate is either broken down into smaller products or joined with other molecules to make a larger product. And once the reaction is complete, the products no longer fit the active site shape. They are released from the enzyme. And here's what's remarkable. The enzyme returns to its original shape, completely unchanged and ready to bind another substrate molecule. And then the whole cycle can repeat thousands of times per second. Now let's move on to enzyme specificity. Having talked about the induced fit model, this brings us to a vital concept, the enzyme specificity. Each enzyme is specific to one particular substrate or a type of substrate. Why? Because the active site must be roughly complementary to the substrate initially. The substrate must induce to the correct shape. Only the right substrate can trigger both these conditions. Think of it this way. A key that fits one lock won't fit a different lock. Similarly, an enzyme that breaks down starch won't break down proteins. The shapes just don't match, and the wrong substrate won't induce the right conformational change. This specificity is determined by the enzyme structure as a protein. The sequence and the arrangement of the amino acids creates that unique active shape and flexibility. We will learn more about the sequence and arrangements of amino acids in the upcoming chapters. We need to now understand what can affect how well enzymes work. Two major factors control enzyme activity are the temperature and the pH. Let's tackle temperature first. At low temperatures, enzyme activity is low. Why? The enzyme and the substrate molecules don't have much kinetic energy. They are moving slowly, so they rarely collide. Fewer collisions means fewer enzyme substrate complexes form, so the reaction rate is slow. But here's the key point. Low temperatures do not denature the enzymes. They just slow them down. As temperature increases from 0 degrees toward the optimum temperature, enzyme activity increases. For most human enzymes, the optimum temperature is around 37 degrees Celsius. That's normal body temperature. Why does the activity increase? Because molecules now have more kinetic energy. They move faster, collide more frequently, and more enzyme substrate complexes form. Now, what happens if it goes beyond the optimum temperature? Here's where the things go wrong. When temperature goes above the optimum, typically like beyond 40 to 50 degrees Celsius for humans, denaturation happens. High temperatures break the bonds holding the enzyme in its specific 3D shape. As the enzyme loses its shape, the active site changes shape too. Remember, the active site needs to be the exact complementary shape to bind to the substrate. When the active site is in the wrong shape, the substrate can no longer bind. No enzyme substrate complexes form, and that means no reaction occurs. Enzyme activity drops rapidly and eventually stops completely. And here's the critical part. Denaturing is irreversible. Once an enzyme gets denatured, it cannot regain its proper shape again. It's permanently damaged. So, on a graph, you see an enzyme activity rising steadily to the optimum, then dropping sharply afterward, creating a characteristic bell shaped curve. The second major factor affecting enzyme activity is the pH. That's how acidic or alkaline a solution is. Different enzymes have different optimum pH values, depending on where they normally work in the body. For most enzymes, the optimum pH is around 7, which is neutral, like pure water. But there are important exceptions, like pepsin and enzymes in the duodenum. We will learn more about this in chapter 7. We will just give you a quick brief here. Pepsin is only found in your stomach. It has an optimum pH of around 2, and that's extremely acidic. Makes sense, right? Your stomach is full of hydrochloric acid. And enzymes in the duodenum works best at a pH of 8 or 9. that's slightly alkaline. So what happens when the pH moves away from the optimum? If pH becomes too high or too low, it affects the bonds holding the amino acid chain together. Remember, enzymes are proteins made of amino acids. These bonds can break, changing the shape of the enzymes including the active site. When the active site shape changes, their substrate can't bind properly, and the enzyme activity decreases. The main difference from temperature: sometimes pH related changes can be reversed if you store the optimum pH quickly enough, but extreme pH values cause permanent denaturation, just like extreme temperatures. The syllabus also requires you to understand how we investigate these enzyme activity. Let's look at a key practical investigation. In this video, we will only talk about investigating temperature effects on enzyme activity. However, you can use a similar experiment to find out the pH effects too. A common experiment involves the investigating how a temperature affects the rate of enzyme control reactions. Here's a typical setup. You might use an enzyme like amylase, which breaks down starch, and test it at different temperatures, say 0 degrees Celsius, 20 degrees Celsius, 30 degrees Celsius, 37 degrees Celsius, 50 degrees Celsius, and 70 degrees Celsius. At each temperature, you would mix the enzyme with its substrate, which is a starch. Time how long it takes for the reaction to complete. You might test for starch using iodine solution. It turns from orange-brown to blue-black when starch is present, as we revised in Chapter 4. The independent variable here is the temperature, and the dependent variable is the rate of reaction. Control variables include concentration of enzyme, concentration of substrate, pH, and the volume of solutions. Your results should show that the reaction rate increases with temperature up to the optimum, then rapidly decreases as the enzyme denatures. And that brings us to the end of Chapter 5, Enzymes. In this chapter, we explored how enzymes act as biological catalysts, speeding up reactions that keep cells and organisms alive. We learned about the lock and key model, the induced fit model, the effect of temperature and pH on enzyme activity, and how enzymes can be denatured when conditions go beyond their optimum range. You also discovered how enzymes are used in metabolic reactions and practical applications. Here's an exam tip. Make sure you can explain how changes in temperature and pH affect enzyme activity and interpret enzyme graphs. These are popular exam questions. Next up, we will move into Chapter 6, where we will look at how plant nutrition works through photosynthesis, one of the most important enzyme-driven processes in nature. If you found this lesson helpful, don't forget to like this video, subscribe, and turn on the notification bell so you never miss the next chapter in our Examify IGCSE Biology Revision Series. You can also join our free WhatsApp community for updates and live sessions. The link's in the description below. And if you're ready to take your revision to the next level, check out our Examify mock papers and topical revision packs designed to help you study smart and score higher. Until next time, keep revising smart, stay curious, and keep exploring the science of life. See you soon in the next video.